Well, hello there. Welcome to the Visual Histology videotape series. I'm Dave Moran, and today's program is on the microscopic anatomy of the cell. Now, I've just been here at the Boulder campus of the University of Colorado, working with this high-voltage electron microscope here, looking at some cells, and every time I look at cells, I'm amazed by their complexity. You know, it's been estimated that each cell has on the order of 10 billion protein molecules in it. That's a lot of molecules for a cell to keep track of. You wonder how the cell can possibly maintain order when everything tends toward disorder. Well, one way in which cells do this is by subdividing their cytoplasm into compartments called organelles. Now, some organelles are surrounded by membranes and others are not. Likewise, some are visible at the level of the light microscope and some are not. Now, you all will be using light microscopes in your study of microscopic anatomy, so what we're going to do in today's program is take you sort of on a journey through the cell with the light microscope, showing you the components that you can see and what they look like. So, let's go to the microscope. What we're looking at here is a diagram of a so-called typical cell, if there is such a thing, shown in your visual histology text atlas. And the arrow is pointing to the outer limit of the cell, which is the cell membrane. It's also called the plasma membrane, sometimes the plasmalemma. And inside the cell we can see several of the major organelles. Here is a mitochondrion, for example, and it's drawn at higher magnification here. And you can see that the mitochondrion consists of two systems of membranes, an outer membrane and then an inner membrane, which is thrown into these folds, which are called cristae. Mitochondria sit right at the limit of resolution of the light microscope, so you have to look very careful to see them with the light microscope. In the electron microscope, however, they're very, very clear. Here we have a system of membranes called the rough-surfaced endoplasmic reticulum. It consists of a series of flat membrane-limited cisternae that have ribosomes on them, and the rough endoplasmic reticulum, sometimes called the rough ER, is responsible for the packaging of proteins, usually for export from the cell, sometimes for use inside the cell. And the rough ER is shown at higher magnification in this diagram over here, and these little dots you see are the ribosomes, the stations of protein assembly, which are on the membranes. In addition to the rough endoplasmic reticulum, you also find the so-called smooth endoplasmic reticulum in cells. It and the rough ER are continuous, but they really are quite different in morphology and often different in function. Here is a high magnification drawing of the smooth ER. It consists of a series of anastomosing tubules. It doesn't have ribosomes on it and serves many functions such as detoxification of poisons in liver cells or steroid metabolism, glycogen metabolism, and that sort of thing. Over here we see some little round spherical membrane limited bodies. These are called lysosomes, shown at higher magnification here. And these lysosomes are filled with a vicious array of hydrolytic enzymes that can dissolve almost anything, like proteins, nucleic acids, polysaccharides and such. And lysosomes sometimes attack foreign materials that come into the cell and eat them up. Sometimes they digest used up intercellular components. Over here above the nucleus we see, and the nucleus of course is right here, right in the center of the cell, and this is the nucleolus here. Above the nucleus we see a system of flat membrane saccules called the Golgi apparatus, shown at higher magnification here. And the Golgi is concerned with the packaging of proteins for export. It wraps them up into these little vesicles, which are then released from the cell. Sometimes they're used inside the cell as well. For example, the Golgi is intimately involved in the production of lysosomes. And the Golgi apparatus is also involved in tacking on carbohydrate moieties to proteins when glycoproteins are made. Also in the cell you will find, like right here, centrioles, usually near the nucleus. And a centriole is shown at higher magnification here. It consists of 9 triplets of microtubules associated in sort of a cartwheel form, shown in cross-section here. And in animal cells, such as human cells, centrioles are seen at the poles of the mitotic spindle. And often centrioles are found at the bases of cilia, in which case they're called basal bodies. And up here, the apical cell membrane is thrown into a lot of folds called microvilli. So this gives you sort of an overview of some of the components that are inside living cells, and what we'll do is look at them with the light microscope in the next image. Here we're looking down on a sheet of mesothelial cells with the light microscope, and this is not a histological section through a tissue at all. This is a case in which a sheet of cells has been spread out over a slide, and we're looking down over the top of them, and the cells have been stained with silver. What you can see here is the outline of each cell. Now, you cannot see the cell membrane with the light microscope, it's too small. But what you can see in this case is the space between adjacent cells, which has been encrusted with heavy metals. So the purpose of showing you this is just to sort of show you the general shape of a series of mesothelial cells spread out on a slide, and all you can see here is the outline of the cell and the nucleus. Now this is very different from the diagram we were looking at, and it points out something very important, that you can't see that many of the cell's organelle systems with the light microscope. But you have to learn to use your imagination. So by looking at a series of images with the light microscope and comparing them with electron images, what we hope to do is sort of build up the capacity for X-ray vision in your mind so you can tell what's really there even though it's kind of blurry with the light microscope. Here we're looking at our sheet of mesothelial cells again, but this time with the Nomarski interference contrast microscope, and that allows us to see contours a little better. So in this case, you can really trace the contour of this cell. Again, we're not looking at the cell membrane, but we're looking at metals taken up by the intercellular space between cells. So you can see that these cells are shaped basically like flat polygons, and you can see the nucleus in the middle. You can see some little threads here which may represent mitochondria, but that's about it. This image is an electron micrograph of a pancreatic acinar cell, which is a cell which is involved with very rapid rates of protein synthesis and secretion. This image comes from figure 1-2 in your visual histology textbook, and the arrow right now is in the nucleus. The nucleolus is here, the rough endoplasmic reticulum is here, secretory vesicles are here, this is a mitochondrion right here, and in particular I want to call your attention to the cell membrane out here. Now this little thin line is the cell membrane, which is the outer limit of the cell, and it's shown at higher magnification here, and what we see here really are two cell membranes, one from this cell here and one from the cell above it, that are next to each other, and between them is the so-called intercellular space. Now in the next image we'll look at this at higher magnification. Here the electron microscope shows us two cell membranes next to each other with the intervening intercellular space. The arrow is pointing at the cell membrane of the lower cell, now it points at the cell membrane of the upper cell, and now the point of the arrow is in the intercellular space. And if you look carefully, you notice that each cell membrane looks like a pair of railroad tracks. There are two dark outer lines and a white clear inner line. This is the characteristic appearance of the cell membrane under the electron microscope. When first seen, this was called the unit membrane because membranes that surround cells look ultrastructurally like the membranes that surround cellular organelles. Biochemically, they can be very different. There's a major take-home point that I want you to think about, and that is that although the cell membrane looks quite similar along its entire circumference, patches of cell membrane can vary dramatically from one area to another. So the homogeneous structure you see under the electron microscope does not give a clue as to the tremendous biochemical diversity manifest by cell membranes at different points along their borders. This is a light microscopic image of the same cells we were just looking at electron microscopically. This is a group of pancreatic exocrine cells. They're also called pancreatic acinar cells, and they're so called because a ball of these cells is organized into a secretory unit called an acinus. In this case, you have a lot of cells like this one here, this one here, this one here, that are all forming these secretory granules that are pouring into the lumen at the center of the acinus. Now this preparation is a light micrograph of a preparation that was prepared as you would prepare specimens for electron microscopy, so a tremendous amount of detail can be seen even at the level of the light microscope. This is not like standard histological preparations, but let's just have a look here. You have the nucleus here, nucleolus here, and these dark streaks are the mitochondria. You can actually see these quite clearly in this preparation. This dark area here is the rough endoplasmic reticulum. These dark little BBs are the secretory granules, sometimes called secretory vesicles, sometimes called secretory droplets, and this is the lumen into which they all pour. Now we're going to concentrate on the nucleus of the cell for a minute, so this is how your typical interphase nucleus looks inside a cell. As you know, nuclei divide. Nuclear division is called mitosis, what we're going to do is have a look at several of the different phases of mitosis in a radically different tissue. What we're looking at here is a really very beautiful light microscopic preparation that very clearly shows all the stages of mitosis or nuclear division. This is taken from an onion root tip, and it's sort of the classical preparation we use to demonstrate mitosis because the cells are organized in a polar fashion in columns, and the mitotic spindles are all oriented in roughly the same direction. Now the arrow here is pointing to the so-called interphase nucleus, and this is a nucleus in which the chromosomes are more or less unwound, the DNA is spun out, and so you see some dark spots here which are nucleoli, and you see some dark regions in the nucleus which are called heterochromatin, and you see some clear regions in the nucleus which are called euchromatin. Now, when mitosis begins, the chromosomes condense, and the first stages of chromosomal condensation are seen here in prophase. The next thing that happens is that the chromosomes align themselves along a metaphase plate, in this stage called metaphase. In the next stage, the chromosomes pull apart, and that is called anaphase. And in the next phase, the two daughter nuclei are formed, and this is called telophase. So mitosis is a very orderly process which ensures that the chromosomal material can be evenly distributed between two daughter cells from one parent cell. We're looking at an onion root tip again, but this time with the Nomarski interference contrast microscope at high magnification. And this particular cell that the arrow is pointing to here is a metaphase cell. And in this case, you can see the chromosomes here and here, but you can also see the mitotic spindle running from here to here. And the mitotic spindle has many spindle fibers in it, to which some of the chromosomes are attached. These spindle fibers are made of cytoplasmic microtubules. Microtubules only measure about 250 angstrom units in diameter, so they're visible with the electron microscope. Individual ones are not visible with a conventional light microscope, but you can see groups of them clumped together to form these so-called spindle fibers in this preparation right here. This is a light microscopic image of a single chromosome. Now, this is not your typical chromosome. Most chromosomes are quite small. This is a giant chromosome taken from the salivary glands of the fruit fly Drosophila. And I show it to you just to show you the characteristic banding patterns that you find on Drosophila chromosomes. The fruit fly has been used for many years as a genetic tool, and one reason it's such a good genetic tool is because it has large chromosomes in which you can see different banding patterns which sometimes reflect different patterns of gene expression. Well, we've just looked at the nucleus and chromosomes, and now we're going to concentrate on the cytoplasm, and to do that we're returning to the pancreatic acinar cells, which we're a little bit familiar with now. The arrow indicates a ball of these acinar cells, but these are stained with a different stain. Beforehand, we were looking at pancreatic acinar cells stained with a stain called toluidine blue. Now we're looking at pancreatic acinar cells stained with hematoxylin and eosin. This particular stain, which is usually called H&E, is the most common stain used for light microscopy. And it's commonly used because it has a very nice characteristic. Some things stain purple with it, some things stain red. In the dye, the hematoxylin is purple and the eosin is red, so certain cell components attract the hematoxylin. Now, hematoxylin is a basic dye, so acidic things in the cell, like, for example, the ribosomes of the rough endoplasmic reticulum, tend to attract it. So in this pancreatic acinar cell, you have lots of rough ER located at the base, and it's going to stain purple with hematoxylin. The secretory granules, which are in the apical part of the cell here, stain red with eosin. They're somewhat basic, and so they attract the eosin, which is an acidic dye. Now, one point of confusion. Things that attract basic dyes are called basophilic, like the rough ER. Things that attract acid dyes, like the secretory granules, are called acidophilic. These terms are used quite often. Now, in order to see these components more clearly, let's look at the same cells with the electron microscope. This electron image here is taken from plate 1-1 in your visual histology text atlas, and it shows a pancreatic acinar cell. The basal pole of the cell is here, the apical pole of the cell is here. And in the basal pole of the cell you can see many cisternae of the rough endoplasmic reticulum, and this is what stains purple with hematoxylin in H&E preparations of these cells. The rough endoplasmic reticulum is encrusted with ribosomes, and the nucleic acids attract the basic dye, which is hematoxylin, which is purple. The secretory granules are here and here and here. These are very acidophilic. These would attract eosin, and hence they'd stain pinkish or red in H&E preparations. And you can see where they're pouring into the lumen right here. Now this is a light microscopic image of a cell that's very different from the pancreatic acinar cell, but still has some of the same components. This is a large motor neuron from the spinal cord, and we're looking at the cell body here. And I'll trace the approximate contours of the cell body with the arrow here. Now, the motor neuron has a large centrally located nucleus here, and it's mostly clear. It's mostly euchromatic. There's a dense nucleolus in the middle, and then you can see some sort of purplish patches. Now, this is stained with toluidine blue. In toluidine blue sections, the rough endoplasmic reticulum stains either blue or purple, and these clumps here are clumps of rough endoplasmic reticulum and free ribosomes. In nerves, they're called the Nissl substance. And out here you can see little piles of dark staining sort of BBs. These are lysosomes that have ingested something from the outside world and have been unable to completely degrade the materials. So these structures are called lipofuscin granules. They're also called wear and tear granules, and they represent lysosomes that have glommed onto something that they cannot digest. In nerve cells, lipofuscin granules or wear and tear granules tend to build up with age. So the more you see, in general, the older the nerve cell. Lysosomes tend to be too small to see with the light microscope, but when they've combined with something indigestible and formed these large complexes, they're quite readily visible. We're going to have a look now at another prominent membrane system in the cytoplasm of cells called the Golgi apparatus. This is an electron image of a Paneth cell. It comes from plate 1-2b in your visual histology textbook, and the Golgi apparatus consists of a series of membranous sacs that are closely associated with one another that give rise to these vesicles that then will form larger secretory vesicles. In this particular case here, in the Paneth cell, these large bodies that are being formed by the coalescence of Golgi vesicles contain lysozyme, which digests bacteria. Now, the Golgi apparatus is very well documented electron microscopically, but is very hard to see light microscopically, so be aware that in special stained preparations, you can see it with the light microscope. More often than not, you cannot. In the next image, we'll have a look at a light microscopic preparation that's been specifically stained for the Golgi apparatus. This is a light microscopic section through the epididymis, which is a tube that leads from the seminiferous tubules in the testis and goes to the vas deferens. It's lined by a pseudostratified columnar epithelium, like this one here. The nuclei are here and here. This is the apical surface of the cells up here. And one characteristic of the epididymal cells is that they have a very well-developed Golgi nuclei, which is above the nucleus. It's in what we call a supranuclear position. And in this section, which has been stained with heavy metals, you see the Golgi apparatus appearing as these large, dense areas. Now, you can't see the membranes for sure, but you can see where the metal has deposited. So this is where the Golgi would be in cells of the epididymis. As you've probably gathered from the images you've seen so far, cells differ radically in size and shape from one another. And here's an odd-looking one. The arrow is pointing to a fat cell here, and it looks like a large soap bubble. One reason for that is that fats are extracted during tissue preparation for light microscopy. This is a fat cell or adipocyte, and what's happened here is that the entire, or almost the entire cytoplasm of the cell has been filled with lipid. The working parts of the cytoplasm have been reduced to this little area you see right here, and the nucleus has been squashed off to one side and just looks like a flat little crescent. So this is how fat cells look in the light microscope, and in this case the cytoplasm has basically replaced itself with its own accumulated product. We're now going to move on and look at specializations of the cell surface. The cell membrane takes many forms, and at the surface of cells it's very often highly modified. And one kind of cell surface modification or specialization that you'll be looking at quite often in your study of microscopic anatomy is the cilium. And when a lot of cilia are together, you very often have an image that looks something like this here. This is the surface of the epithelium of the trachea. It has a whole group of cilia. The cilia look kind of like the bristles of a brush. The cilia would extend from the epithelial surface here right out to about here. Each cilium is a little too small to see with the light microscope. It measures about 2/10 of a micrometer in diameter, which is the resolving power of the light microscope. But let's have a look at another image and see how these cilia look at higher magnification. This is a light microscopic image of an H&E stained preparation of a section through the bronchus of a monkey. It's shown at slightly higher magnification than the previous section, and so the cilia, which you see here, stand out a little more clearly. You can see them almost presenting an image like the individual bristles of a brush. They extend from the surface here right out into a layer of mucus, which they propel. In this case, the epithelium extends from here to here. It sits on a well-developed basement membrane here and is supported by some loose connective tissue here called the lamina propria. There are several different kinds of cells lined up next to one another in this ciliated pseudostratified columnar epithelium, such as mucus-secreting goblet cells here and ciliated cells here. Although cilia can be somewhat difficult to resolve on an individual basis with the light microscope, they're very easy to see in the electron microscope. These are several ciliated cells from the epithelium lining the human nose here. This is the surface of the cells, and from the cell surface extends these cell surface specializations, the cilia, here, and each cell has a large number of them. The ciliary shaft, as I mentioned, is 2/10 of a micrometer in diameter, and it's very easy to see electron microscopically. When you look at it in cross-section, the ciliary shaft has a very specific ultrastructure. This is an electron image at high magnification of a cross-section through one of these cilia on the respiratory epithelium of the nose, and the arrow is pointing to the cell membrane that surrounds the cilium. Remember, the cilium is an extension of the cell surface, so it's coated by the cell membrane. You can see that sort of railroad track unit membrane appearance of the cell membrane here. The cilium is supported by cytoskeletal elements which taken together are called the axoneme. And the axoneme consists of, among other things, 9 doublets of cytoplasmic microtubules here, each of which has a pair of arms called dynein arms. Now these dynein arms have an ATPase in them called dynein ATPase, and they can move back and forth like little ratchets, which cause the adjacent doublets to slide relative to one another. These doublets are constrained from moving very far by these structures here, which are called radial links. Consequently, when the doublets slide, the sliding motion is transduced into a bend, and the cilium bends and has an active stroke. Remember, these cilia are not static. They're highly modal organelles. In single-celled animals and plants, they can propel the organism at a rapid rate. On an epithelium, like the epithelium we've looked at, they can move mucus along their surface very fast. So cilia are highly motile organelles. They're used to move either whole cells or material across the surface of cells, and have this characteristic so-called 9+2 substructure in which you have 9 outer doublets surrounding 2 central microtubules, which is called the central pair. There's another kind of very important cell surface specialization called the microvillus, and the plural is microvilli. This is a light micrograph of an intestinal villus, and each of the intestinal epithelial cells here is covered by a group of microvilli. Now, they're too small to individually resolve with the light microscope, but taken together they form this structure here, which is called the brush border, which is a whole bunch of microvilli arranged in a row. Now, let's have a look at this at higher magnification on the next slide. This is a light microscopic image of the same specimen we were looking at, but taken at higher magnification. And right here, the arrow is pointing to the brush border, which consists of a series of parallel microvilli. You can see the base of the brush border here, the apex of it here, and it's this sort of pink staining area that runs along the cell surface. Now, in addition, there's a special kind of cell that we see here, the goblet cell, which is a mucus factory. We're going to take a look now at several different kinds of cells that do different things to underscore the point that cells can look very different from one another and that their morphology very often connotes their function. So this is how a goblet cell looks. It looks pale. Here's another one up here because much of the mucus has been extracted by specimen preparation, and it's shaped somewhat like a goblet, and its morphology can be seen even better with the electron microscope, as on the next image. This is an electron micrograph taken from plate 1-3 of your visual histology text atlas, and the arrow is in one of the intestinal epithelial cells, the kind we just looked at, and you can see the individual microvilli right here. This is how they look in the electron microscope. They have a diameter of about 1/10 of a micrometer, so they're too small to see with the light microscope, but they're very readily evident in the electron microscope. Now, next to this intestinal epithelial cell is a cell that looks radically different. This is a goblet cell here, which is a mucus factory, and the apical pole of the cell is packed with these mucus droplets right here, and they have sort of smashed the cytoplasm against the basal pole of the cell. You can see lots of rough endoplasmic reticulum here. You can see a dark staining nucleus, and you can see some of the Golgi apparatus here. The take-home point here is that you've got a cell that makes a lot of mucus. The mucus piles up at the top. It endows the cell with this shape, which is somewhat like a goblet, hence the name. And just keep this cell shape and function in mind when we look at some of the other cells as we go through a series of images. The next cell we're going to consider is the osteocyte. Now, osteocytes are bone cells, and what you see here is a light microscopic image taken with the Nemarsky interference contrast microscope of a piece of bone that's been ground wafer thin on a special grinder. So the living tissues are not here. The calcified part of the bone is here. The spaces have been filled with India ink. And what I want to call your attention to with the arrow here is this dark space here filled with India ink. This is the space that formerly was occupied by an osteocyte. Now keep this in mind while we show you an electron image of an osteocyte. Now here is an osteocyte when it's seen with the electron microscope. This comes from plate 1-5 from your visual histology textbook, and you can see right here that this osteocyte is really a tiny cell. Nucleus is here. There's only sort of a remnant of the cytoplasm. There are some osteocyte processes that extend out through tunnels in the bone here, and this is all solid bone. The reason for showing you this image is that the osteocyte is a quiescent cell. It isn't really doing a lot. It's necessary to maintain the bone, but it's not being secretory at any great rate of speed. Consequently, its cytoplasm is reduced. So quiescent cells tend to be really quite small and appear to be relatively undeveloped compared to other cells that are active, say for example protein factories in the pancreatic acinar cell or goblet cells in the intestine. And here are some cells that look completely different from any that we've seen. In some cases, the cell will basically fill its cytoplasm with its own product, and that product will perform the function of the cell in later life. Classic example is the red blood cell. And these cells here, which look like sausages in this case, or donuts over here, are red blood cells that are passing through a capillary whose walls are here and here. The red blood cell is basically a large crystal of hemoglobin. It's replaced its entire cytoplasm with its own secretory product, which is hemoglobin, which then goes on to bind oxygen and is very important in the physiology of respiration. Other cells, such as the muscle cells shown here, have filled their cytoplasm with filaments that participate in muscular contraction. So in this case, the cell has filled itself with a product which it uses to perform its own function. So muscle cells look very different from blood cells. Blood cells look very different from bone cells. These look very different from the mucus factory of the goblet cell, and all of them look quite different from the protein factory, which is the pancreatic acinar cell. Now there's one very important thing to remember, and that is that not only do different cells that perform different functions look different, but a single cell can look radically different at different stages of its own life. What we've got here are electron images of the same kind of cell at different stages of life. On the right-hand side we have a B lymphocyte. This is inside a capillary. It's a circulating leukocyte, a white blood cell. And on the left we have an electron image of a plasma cell. A plasma cell is an antibody-secreting cell. It's not in the bloodstream. It's located in the connective tissues, and just by inspection you can see a radical difference between the cytoplasmic components of these two cells. This B lymphocyte here has a very limited cytoplasm. You can see a centriole here, a mitochondrion here. Compare that with the cytoplasm of the plasma cell over here. Tremendously well-developed rough endoplasmic reticulum. Why is it there? Well, these plasma cells, situated in the connective tissues, secrete antibodies which are proteinaceous. Consequently, the cytoplasm is going to be loaded up with the rough endoplasmic reticulum, which is specifically designed to manufacture proteins for export. The take-home point here is this is the same kind of cell at different stages of life. This is the young circulating B lymphocyte here and the mature plasma cell into which the B lymphocyte grows and differentiates with time. Now I think you've got a fairly good grasp of the light microscopic and electron microscopic appearance of cells. Let's let you test your knowledge here with a practical examination, which will come up next. Now we're going to start a practical exam, and I'll tell you what the format's going to be. It'll be quite simple. I'm going to point to a particular structure on the screen with an arrow and ask you what it is, and then I'll give you 10 seconds to think about what it is. Now 10 seconds isn't an awful long time. If you need more time, just stop the tape. And then at the end of 10 seconds I'll tell you what it is, and if you assume pencil and paper and write these things down as we go, you'll have a notion of how much you've digested. So here we go. Question number 1. What is this structure the arrow is pointing to here? And it runs right along the surface. And the answer is the brush border. This structure is called the brush border, and it consists of microvilli. Now, question number 2 on the same image. What kind of cell is the arrow sitting in right here? If you said a goblet cell, you're right. This is a mucus-secreting goblet cell, and in this case, it's located in the intestinal epithelium. Question number 3. What is the structure that the tip of the arrow is touching right here? If you said the nucleolus, you're absolutely right. The arrow is pointing to the nucleolus. Here is the nucleus, and the nucleolus is this dent staining body here. In other cells, for example, there's one here. Let's go on to the next question. What are these little dark structures here that look like sort of purplish black circles? Another one here, here. What are these called? These are secretory granules. This particular image is of the acinus of a pancreas. It's filled with pancreatic acinar cells, and the rough endoplasmic reticulum down here is manufacturing the secretory granules, which are up in here. Question number 5. In what stage of mitosis is this cell right here? If you said anaphase, you're absolutely right. Remember, this is the section of the onion root tip which has prophase here, metaphase here, anaphase here, and telophase here. And anaphase is the stage at which the chromosomes progress towards the opposite poles. Here we have a bunch of black or dark blue spherical cytoplasmic inclusions. What are these called? The arrow's pointing to one right here. And the answer? These are lipofuscin granules. They're also called wear and tear granules. And this particular cell is a motor neuron from the spinal cord. Question number 7. What are these structures right here? If you said cilia, you're absolutely right. These are cilia. They're on the surface of the epithelium of the respiratory tract. And remember, cilia are fine extensions of the cell surface that are highly motile. Question number 8. What kind of cell is the arrow sitting in right here? That is a fat cell, or adipocyte. Here's another one over here, and another one over here. In the light microscope, in standard H&E preparations, these fat cells tend to look sort of like soap bubbles. What is the arrow sitting in and pointing to right here? You can see a whole series of sort of thin lines that are parallel. What system are these lines all part of? If you said the rough endoplasmic reticulum, or rough ER, you're absolutely right. This is an electron micrograph of a pancreatic acinar cell, and here in the basal pole of the cell we have a very well-developed system of cisternae of the rough endoplasmic reticulum. Now, in this same image, there are some other organelles, like this one here, for example. What is the name of the organelle being pointed to by the arrow right here? And the answer is a mitochondrion. There are a number of mitochondria in this cell. There's one here, one here. So if you said mitochondrion, you're absolutely right. Well, this concludes our videotape on the cell. It's been a pleasure being with you. And if there are any points that need clarification, just run through the tape and have a look at it again.